Hey everyone, and welcome to today's Explainer. I'm absolutely thrilled to have you here, because today we're tackling a seriously fascinating theme. We're looking at how massive, incredibly complex systems handle sudden disruptions. Think about it, whether we're talking about human societies, biological organisms, or even highly technical synthetic brains, every single system operates on a baseline. But what actually happens when you introduce a massive shock, an anomaly, or a rapid shift into that baseline? How does the system adapt, reroute, or just totally transform? Let's find out.
Okay, let's dive into this. Here is our macro to micro roadmap for today. 1. Global science policy shifts. 2. Avian color variations. And 3. Bifurcating neural networks.
All right, starting with Section 1, Global Science Policy Shifts and Navigating New Political Realities.
So right now, our human global systems are experiencing a wave of massive political and administrative shocks. And you really have to wonder, how does international science, which relies on stability, adapt when its foundational funding and policy structures face rapid, totally unprecedented changes? Science is basically this giant, globally connected network. And right now, the rules feeding into that network are shifting dramatically in the United States.
Currently, we're seeing several major rapid disruptions hitting the federal level. For instance, the Office of Management and Budget, the OMB, recently sent out a 30-minute survey to foreign researchers who receive U.S. funding. It's probing whether their work actually aligns with new administration priorities, asking about things like national security and foreign funding. At the exact same time, the newly formed DOG initiative has mandated massive reductions in force, or RIFs, at federal science agencies like the EPA. And on top of all that, the National Institutes of Health has seen its recent scientific integrity policies entirely rescinded because they included diversity, equity, and inclusion requirements. So we are looking at significant, sudden variations to the baseline rules of federal science.
And this quote right here perfectly illustrates the ripple effect this has across the globe. Vicky Thompson, who represents Australia's leading research universities, pointed out just how alarming these new surveys are. She stated, and this is a direct quote, that for every one of our members, the U.S. is the largest research partner by far. In fact, because of these sudden administrative hurdles, some Australian universities have already lost U.S. funding for their research. It's a textbook systemic shock, a disruption in one node, the U.S., immediately impacting nodes all over the world.
Which brings us to how the rest of the system reacts. It's fascinating to compare. On one side, you have the U.S. policy shifts bringing heightened border scrutiny. For example, a French space scientist was recently denied entry. The French government claimed it was due to political text messages, while U.S. Homeland Security stated it was actually a violation of a non-disclosure agreement regarding confidential lab information. But regardless of the why, the friction at the border is undeniably increasing. Now, look at the other side. The European system is actively stepping in to absorb this disruption. France and the Netherlands are creating brand new funds specifically timed and designed to attract international STEM talent that might be displaced by the U.S. layoffs. It's a perfect example of a complex system rebalancing itself. When one part of the network pushes talent away, another part instantly rolls out the welcome mat.
Moving on to Section 2, Avian color variations and disruptions in the biological code.
So we're shifting gears from human policy over to nature, but the concept is exactly the same. Just as new policies alter the landscape of global science, when biological code encounters a mutation or a developmental defect, the physical outcome of that organism completely transforms. To really visualize this, we're going to use bird plumage as our guide to see exactly how nature processes anomalies.
First up is albinism. Now you've probably heard of this one. It's a genetic mutation that actively interferes with the production of the pigment melanin. But here's what's wild. Unlike humans, animals often have other pigments besides melanin. So a true albino bird isn't necessarily totally white. They might actually retain some species-specific patterns, like wing bars. However, this mutation is a severe disruption to the bird's system. Without melanin, their feathers literally lack structural support and wear out super quickly. Plus, without protective pigments in their eyes, they can easily go blind.
Next, we have leucism. Now, a lot of people mix this up with albinism, but it's totally different. Leucism isn't a genetic mutation of the melanin code itself. Instead, it's a defect that happens during development in the pigment cells. This results in either an overall paleness or irregular white patches all over the bird, something birders call being piebald. And crucially, because eye development happens completely separately from feather development in the biological sequence, leucistic birds retain perfectly normal eye color.
Now, if we jump to the total opposite end of the spectrum, we find melanism. This is an excess of dark pigmentation, and it can be triggered by a genetic mutation or sometimes even just a specific diet. What's really interesting here is that some complex biological systems have actually integrated this anomaly into their baseline over time. The red-tailed hawk, for example, has a naturally occurring melanic form or morph that is just a totally standard variation of the species today.
And finally, we have xanthochrosum and erythrism. Yeah, try saying those 5 times fast. These are conditions that cause unusually yellow or orange feathers, which you'll often see in house finches, or reddish rufous feathers, which are super common in the eastern screech owl. Again, these are systemic variations caused by genetic shifts or environmental inputs like diet.
Now, what's really interesting about this slide is how crucial these differences are in the field. If you're out birding and you spot a systemic disruption in the wild, you need to know the markers. True albinism is a full melanin failure, weak feathers, pale eyes. Partial leucism is a developmental defect, piebald catches but totally normal eyes. It's essentially the same basic disruption, a lack of pigment, but the underlying biological mechanism processing that shock is entirely different.
All right, let's hit section 3, bifurcating neural networks and the synthetic brain.
Okay, so we've looked at human societies, we've looked at biological organisms, now let's get into computational systems. The big question we're asking here is, how does a synthetic brain process and store associative memory? In your standard computer, everything's rigid. It's binary, just ones and zeros. But the human brain doesn't work like that at all. We rely on complex, continuous associations. So how on earth do we recreate that in an artificial neural network? Well, the answer might just lie in introducing a little controlled chaos. Think about the way you recognize an object, like a coin. Our brains don't just store that data as a flat binary snapshot. We store rich, continuous analog associations. We recognize that coin from different angles or under completely different lighting. To give a synthetic brain this kind of capability, scientists are exploring models that don't just flip on or off, but can actually hold a continuous phase of memory.
So the crucial point is understanding the mechanism driving this, which is known as the integrate and fire neuron. It's pretty straightforward when you break it down. The neuron's internal potential rises as it gets input. Then, once it hits a specific threshold, boom, it triggers a spike, it fires. And immediately after, it drops instantly back down to a relaxation level. It's just this continuous, rhythmic cycle of building up energy and releasing it.
But here is where we intentionally cause a systemic disruption. What happens if scientists introduce a constant driving signal? Imagine it like an unrelenting external metronome just ticking away at a specific frequency. It completely disrupts that normal, peaceful, integrate and fire pattern. It actually forces the artificial neuron into a chaotic, bifurcating state where its firing times start to split and shift wildly out of control.
When you map this out visually, it looks like literal chaos. You get these incredibly dense bifurcation diagrams where the firing times are just splintering off in seemingly random directions under the pressure of that metronome. But guys, here's the absolute wildest part. This chaos is not a system failure. By pushing the network into this bifurcated state, it starts acting as an amplitude-to-phase converter. That means it translates the raw strength of an incoming signal into a specific, continuous rhythm. That chaotic firing actually allows the network to lock onto and store those complex analog memories we talked about earlier.
So to summarize these really dense, highly technical breakthroughs, injecting controlled chaos into the system is exactly what enables analog memory storage. The networks essentially learn to convert the amplitude of an input into a distinct phase of firing. And ultimately, this points researchers toward a massive unified framework called bidirectional wave transcription. In plain English, it means that by actively disrupting the baseline system, scientists have unlocked a mind-blowing capacity for associative memory, all without having to expensively add millions of new artificial neurons. It's brilliant.
Which brings us to the end of our journey today, and it leaves us with one seriously profound question to think about. Whether we're looking at international science policies scrambling to react to political shocks, biological codes mutating to create totally new bird plumage, or synthetic brains harnessing mathematical chaos to unlock memory, are complex systems ultimately defined by their baseline rules, or are they actually defined by the anomalies and variations that disrupt them? Thank you so much for learning with me today, and keep questioning the systems around you.